All right, so again, my name is Sato. I'm the Chief Strategy Officer of Adimab. I'm actually, last week was my 1-year anniversary at Adimab, and if you're not familiar with Adimab, Adimab is relatively, we've been around for about 17 years, and so in the history of Adimab, 1 year is actually a pretty short period of time in the overall history of Adimab. And so for today, I'm going to give you an overview of all the amazing things that I've learned in my 1-year stint at Adimab. So if you're not familiar with Adimab, Adimab is a premier therapeutic antibody discovery company. We're based in Lebanon, New Hampshire, and we also have a small site in Mountain View, California, which is the site that I'm based in. We're a privately owned company, and we were founded in 2007, as I mentioned before in my previous slide. We're really focused in discovering therapeutic antibodies, and since we were founded in 2007, we've worked with over 130 different partners, so really amazing validation of the overall platform. We have no internal pipeline of our own. We've really focused on making sure that our partners are successful as they move into the clinic. In working with those 130 partners, they've advanced over 80 molecules into the clinic. Again, amazing validation of the platform. And of those 80 molecules, 5 of them have advanced onto the market today. So as I mentioned, I'm pretty new to Adimab. And so when I joined the company, I was really reminded of this book that I read as a child, which was Charlie and the Chocolate Factory. And the reason why I was reminded of this book is that when Charlie got this amazing opportunity to tour Willy Wonka's chocolate factory, he was just blown away by all these amazing inventions that he got to see when he got to tour Willy Wonka's chocolate factory. And so similar to Charlie, I was just blown away by all these amazing inventions and technologies that I got to see in my first year at the company. Shown here on the right is a map of the factory that Charlie got to tour during his day at the factory at Willy Wonka's chocolate factory. And so similar to this map, I'm going to walk you through a map of the Adimab factory. It's kind of a template for my talk today. So these are basically 10 different rooms in the Adimab factory that I'm going to walk you through as a way to kind of give you an overview of all of the technologies and all the amazing things that I learned in my first year at the company. And then you can follow this chevron at the bottom to kind of walk you through those 10 rooms in the overall factory. Okay, so let's start off in the first room, which is our full IgG and HCAb workflow. So these are our synthetic libraries, which are in our proprietary yeast strain, and we have both IgG as well as HCAb libraries. So in terms of our IgG libraries, we have both kappa as well as lambda libraries. We also have a common light chain library as well. For our IgG libraries, we have, which are primarily germline based, we have over 20 different heavy chain germlines as part of our IgG-based libraries. And we also have, for our kappa and lambda libraries, we also incorporate over 10 different kappa and lambda germlines as well. For all of our IgG libraries as well as our HCAb libraries, we also have over 10 to the 10th overall diversity incorporated into these libraries. So amazing diversity, even though we're using yeast display as our primary method of creating our libraries. So we heard in the previous presentation that sometimes creating highly diverse libraries in yeast is difficult, but that's really enabled by our proprietary yeast strain, which allows us to create these highly diverse libraries in our strain. And as I mentioned, we have over 20 different heavy chain germlines and over 10 different kappa and lambda germlines as part of our IgG libraries. I'll spend a separate room in the presentation talking about our HCAb library, and I won't talk about it on this specific slide. Okay, so moving more into the overall design and how we incorporate it into our yeast strain. As I mentioned, all of our synthetic libraries are in silico designed. We incorporate both developability as well as immunogenicity considerations as we design these libraries. We've been redesigning them year after year over our 17-year history. As I mentioned, we also have been, these libraries are heavily germline based as well, with most of the diversity in heavy chain CDR3, with some diversity in light chain CDR3 for the IgG libraries. As we discussed in the previous slide, also, the bulk of the overall diversity of the libraries is on the order of 10 to the 10th. But when we synthesize them, we actually synthesize them on the order of 10 to the 12th. The reason why that is important is that when we make each of these individual libraries, we actually make multiple copies of them. And so that if different partners come to us with the same target, we can actually pull different versions of each of these libraries so that we never have to select the same version of these libraries against the same target twice for different partners. We just heard in the previous talk about not delivering the same sequence twice for different partners. That's how we can make sure that we never have to do that. So we never have to actually go through gatekeeping because we have these different versions of each of the different libraries I talked about in the previous slide. And that's because we synthesize on the order of 10 to the 12th. Another really big differentiator for our libraries is that we actually display the full modality on the surface of our yeast. So we're not displaying a fragment of the antibody on the surface of the yeast. We're actually presenting the full IgG or the full HCAb on the surface of the yeast, which I actually find is really differentiated about our platform in that we're actually displaying the final modality that we're actually providing back to the partner rather than just a fragment of the overall antibody. Okay, so we talked a lot about the overall design and how we build the libraries. How do we actually then implement and discover the antibodies from these synthetically designed antibody libraries? We then take the biotinylated target, we mix it with the libraries, and then we use a combination of max and fax-based selection to then extract out those antibodies. Of course, we can use both, sort for both expression as well as antigen binding. And then we do a series of up to 4 or 5 rounds of selection to pull out those antibodies. Of course, we can drive for higher affinity binding to those targets, but we also can, of course, select out for certain specificities as we're selecting for those antibodies. Adimab also historically came about with our polyspecificity reagent, which we can use to deselect for any antibodies that have poor developability properties, and we can do that by taking a backgate in our selections to get rid of any antibodies that have poor developability. Another great attribute of our yeast is that in the later rounds of our selection, we can pick individual clones and switch from presentation to secretion. And so once we pick those clones, we can then express and purify the antibodies from those soups and then characterize them for kinetics, developability, epitope binding, cell binding, and put them through rigorous characterization. Can we go back one? Actually, can you go back one slide? Go back once, sorry. Okay, great, thanks. And again, we utilize this same workflow for both the IgG as well as HCAb workflows. And during that primary delivery, which it takes about 3 to 4 months, during the later rounds of selection, we also do what's called a batch shuffle process. For our IgG workflow, typically what we would do is what we call a light chain batch shuffle. Now, as I mentioned, the bulk of our diversity is in heavy chain CDR3. So what we would typically do is we would do a light chain batch shuffle to allow the heavy chain CDR3s to see additional light chains during that process. For our HCAb library, typically what we would do is do a heavy chain CDR1-2 batch shuffle to allow those heavy chain CDR3s to see additional heavy chain CDR1s and 2s. Shown on the right is just some typical affinities that we see during that overall primary delivery process. And as you can see, typically what we see are affinities in the range of single-digit to maybe triple-digit nanomolar affinities in that primary delivery. And typically we deliver in the range of 90 to 96 antibodies back to the partner. What we're trying to do in this initial primary discovery delivery is deliver back to our partner a panel of antibodies that hit a wide range of different epitopes. We're not really trying to drive towards high affinity in this primary discovery step. We're really trying to drive towards higher epitopic breadth in this primary discovery step. Because as As we want to drive towards later on in the overall process, we can actually drive down towards higher affinity as we try to optimize these antibodies in later rounds of the discovery process. So as we then deliver those 96 antibodies to the partner can then assess them for overall function. And during that process, they can come back to us and then tell us which antibodies have the right function against that specific target. And then, if needed, Adimab can then do additional rounds of antibody optimization. One great power that I see in the Adimab system is that we can actually do multiple antibodies in parallel for antibody optimization, and that we can actually optimize up to 6 to 8 antibodies in parallel using our proprietary yeast in our system. And we can optimize them for not only affinity, but also specificity and developability if needed. And how do we do that? Typically, we might, for example, focus in on the heavy chain CDR1 and 2, utilizing pre-made cassettes, and then maybe we might focus in on, say, for example, the light chain in CDR1 and 2 and light chain CDR3. And we would utilize that same FACS-based workflow that I showed in the previous slide. And then shown on this slide are some affinity increases that we might potentially see through that affinity maturation process, where the open circles of the same color are the parental antibody, and then the filled circles of the same color are the variants derived from that parental antibody. And as you can see, we typically can see upwards of maybe 1 order of magnitude in overall affinity increases during this affinity maturation process, sometimes 2. Okay, so as I promised, in the next room in the overall Adimab factory tour, I'm going to dive into our HCAB library design. Unlike our IgG libraries where we have multiple heavy chain germlines, for our HCAB library design, we focus primarily on VH3-23 as our base germline framework. But we incorporate multiple tetrads into Framework 2. We incorporate 5 different tetrads into Framework 2 as part of our overall design. In our future design, we're actually going to incorporate an additional tetrad into our HCAB design. Shown here on our right, you can also see the length diversity in heavy chain CDR3, and as you can see, we have a slightly longer heavy chain CDR3 diversity relative to human heavy chain CDR3 in our IgG-based library. We do incorporate some human heavy chain CDR3 diversity in our HCAB library, as well as some camelid-based diversity in our HCAB library. And then also similar to our IgG library, we do synthesize this library on order of the 10 to the 12th and make multiple copies of it so that if different partners come to us with the same target, we can pull different copies of it and use different versions of the library for different partners so that we never deliver the same sequence twice. Okay, moving on to the third room in the Adimab factory is, what if a partner comes to us for either humanization and or optimization of a sequence not derived from our libraries? We can easily do that. We have in-house software for doing humanization, where we can put those sequences into our in-house software and rapidly do humanization, where this software gives us a number of different variants, where it gives us, where we can input the number of allowed amino acids in both the heavy chain as well as light chain framework. We can rapidly make those sequences, make and test them, and then that gives us a number of humanized variants to move forward with into antibody optimization. And then once we have those humanized variants, we can then move forward into antibody optimization using approaches very similar to, as I talked about before, in antibody optimization of sequences derived from our own libraries. So shown here is one approach that we might use to do that, where we take our input antibody and we make 3 different libraries, where, for example, we might make a heavy chain library, a light chain library, and potentially if the antibody has some issues with some liabilities, we might actually make a library focused on fixing some of those liabilities and select all 3 of those libraries in parallel. And then move forward all those selections together into one combined library in cycle 2, and then move forward all those improved progeny in the combined library. And then finally, any liability fixes that come forward in that liability fix library, we can combine them forward in those improved progeny. Shown here is a case study where we actually did exactly what I showed in that previous slide, where we took 3 input parents that were humanized. Shown on the left axis are the affinities of the input parents as well as the variants. What you want to look at are the symbols as well as the colors of the parents, and then you can track the variants by tracking the same symbol as you move down in the graph. Shown in the x-axis is the Hick profile of each of the parents as well as the variants. And what you can see is that the variants improve in overall affinity as well as improve in overall Hick profile as we do the affinity maturation. So we see dramatic improvements in overall affinity as well as improvements in overall developability of variants. Okay, so next room in the overall Adimab factory is our leadership in developability. Early on in the history of Adimab, we focused heavily in developability because we knew that developability was going to be crucial to the overall success of our company. And as you may know, there has been no issues with any antibody that's gone into the clinic derived from our overall platform, and that's because we have focused so heavily in developability. What I've learned since coming into the company is that we do a lot of pre-work and a lot of work during our projects to predict the developability of antibodies through in silico-based approaches, and also we follow that up through a lot of experimental testing of antibodies derived from our platform as well. And we continue to do a lot of technology development in this area, and we also have a lot of data derived from our platform, so we're continually coming up with better and better models for predicting developability as part of our overall platform. Okay, so next room in the overall Adimab factory is our leadership and our ability to do in vivo antibody discovery, which was something surprising to me. I already knew that we had these amazing synthetic libraries, but actually our ability to discover antibodies from in vivo sources was a really mind-blowing thing that I learned upon coming into the company. And this idea that we can actually immunize either mice or llamas, and then actually take the output from these in vivo sources and then port that into our yeast, and then use the yeast as a source to actually do single B-cell cloning was an amazing technology advance that I saw. One really cool thing that I saw is that we can actually capture upwards of 90% of the overall repertoire during this single B-cell cloning event. So unlike a lot of other single B-cell cloning technologies where sometimes you lose a lot of the repertoire doing that single B-cell cloning event, we actually can capture a lot of the repertoire through the use of our yeast and that PCR event. We also have access to the Ablexis transgenic mice as well. So not only can we immunize wild-type mice and llamas, we can also immunize Ablexis humanized mice as well, and use that as a way to capture that diversity through that workflow that I talked about before, either doing single well-based cloning or fax-based cloning using the same workflow that I showed before. Okay, so next room that I want to talk about is our ability to do protein engineering, and more specifically, TCR engineering. Historically, in my past, doing TCR engineering has been very difficult to do in phage display. I mean, it's actually been something that I've actually shied away from because it's been incredibly difficult to do. But what Adimab's been able to do is kind of trick the TCR to basically present much like an antibody. So shown here on the right is where we've actually fused the TCR to an FC, constrained it with a disulfide, and then sometimes we make some mutations in the TCR, and then through that event, we can actually present it on the surface of our yeast, much like an IgG. And then when we can do that, we can build a library around it and then engineer it to bind with higher affinity to its specific HLA, MHC HLA class 1, or potentially in the future a class 2. Shown here in this graph, you can see that in the red dot is the specific TCR. You can see it binds with almost negligible affinity to its HLA class 1, but through multiple cycles of affinity maturation, you can see that we're able to get down into the picomolar range through doing affinity maturation in our yeast. We also made a deep mutational scanning library in a single round of maturation, and we're also able to get down into that picomolar range. And then shown here on this right-hand graph, we actually can show that we only need to make about 9 to 10 different mutations to get down into that range. We also show in that blue dot a control where another company actually affinity matured this same TCR, and they actually required upwards of 14 or 15 different mutations to get down to that same range. So using our approach, we actually were able to use less mutations to get down into that picomolar range. All right, next room in the overall Adimab factory tour is our ability to do general protein engineering. Similar to the TCR example, if we can display it on the surface of our yeast, we can engineer it. So in this case, we had a protein protein receptor that bound 2 different proteins, and we wanted to engineer a way binding to one of those specific proteins. We just made a library around it, and then we were able to deselect away from one of those specific proteins, and we were able to quite easily do that through the course of selection in our yeast. All right, so the next 2 rooms that I'm going to talk about are add-on technologies that we offer in a non-exclusive way that allows us to create higher order, more complex biologics in our platform. And the first one are mutations that allow us to create bispecifics and multispecifics in our technology. We offer these both as a non-exclusive license back to our partners, but we also can use them as part of a funded discovery project with our partners as well. And these include both heavy chain mutations, as well as heavy chain, light chain mutations to create higher order bispecifics and multispecifics. We also have mutations that allow us to do chain exchange screening as well, where we can do rapid prototyping, rapid screening to create bispecifics and multispecifics. And utilizing all of these technologies allows us great diversity and flexibility in this field, where we can make all kinds of different bispecifics and multispecifics in multiple formats. All right, so stepping into the heavy chain, light chain pairing mutations, we have multiple mutation sets that we can utilize to create all kinds of different pairings for both kappa as well as lambda Fab regions in your bispecific and multispecific. So we have mutations between CH1 and constant kappa, CH1 and constant lambda, as well as mutations between CH1 and C kappa. All of these mutations are in the constant regions of these Fab regions. None of them are in the FV region, so they're very plug and play within the overall Fab region. And we don't see any issues with loss of affinity in the FV region. And we actually published all of this data recently in MABS or earlier this year, so I refer you to this publication if you want more information. In terms of our heavy chain mutations, we have 2 different sets. I just briefly talked about our chain exchange mutations, which allows us to, using an in vitro process, create bispecifics and multispecifics really easily as part of a screening process. But we also have our own version of knob and hole, which we can use for scale-up to create bispecifics and multispecifics in a cell line-based or transient-based process as well. Once we home in on a specific bispecific or multispecific after we do the chain exchange process. Okay, last room I'll talk about in terms of our overall technology non-exclusive offering is our CD3 offering, which complements really nicely the bispecific, multispecific technology I just talked about. So we also have our own CD3 IgG binder as well, which we can then utilize to create T-cell engagers, bispecific T-cell engagers as well. And so shown here on the left is we have a CD3 binder that we've then created a whole series of different affinities. This CD3 binder has a wide range of different affinities from very weak to very high affinity into the picomolar range, and has human cyno cross-reactivity. Shown here on the right, you can see that we've taken 2 of these CD3 binders and created a 1+1 bispecific with a CD20 binder, And you can see really nice killing in this killing assay that it compares really nicely with the Genentech CD3 binder in this same format. And so having this wide range of different affinities is really nice because you can basically match it really nicely with your specific tumor antigen binder as you're building your bispecific T-cell engager. In addition to having a CD3 IgG binder, we also have a CD3 HCAB or VHH binder as well. So similar to the IgG binder, we have a wide range of different affinities. This HCAB binder also has really fantastic developability, binds human T cells really well. You can see on the left, because we have a wide range of different affinities, they also kill with different degrees when we pair it with CD20 in a 1+1 format. But unlike the IgG binder, where we have human cyno cross-reactivity, most of these HCAB binders actually bind an alternate epitope. They actually bind the UCHT1 epitope. And so because of that, all of these antibodies actually only have human-specific binding. They don't have human cyno cross-reactivity. And I didn't mention on the previous slide, all those IgG binders that I talked about before bind the N-terminal domain of CD3 epsilon. Okay, actually, can you go back one slide? Sorry. Great, okay. Last but not least, last one I'll talk about is our people, which I find to be an amazing strength of Adimab. As I mentioned, Adimab is a 17-year-old company, and because we've been around so long, we have a lot of people that have also been around at the company for a long time as well. And we have a lot of experience that we bring to the table. We have a lot of PhD antibody engineering scientists, as well as protein biochemists, computational biologists. A lot of people have excellent experience in high-throughput antibody expression, as well as software engineering. And every project that we do for our partners, we bring this deep experience to the table. So I really appreciate, and I've learned from all of these people that I've worked with in this last year, and every partner that works with us really appreciates this deep experience that we bring to the table for every project that we do for our partners. So I just wanted to end with that overall strength of our Adimab platform. Okay, so just in closing, Adimab is a premier therapeutic antibody discovery company with over 80 drugs in the clinic and 5 commercial products derived from our platform. We're experts in developability and have a full stack of antibody discovery libraries and are really experts in antibody optimization as well. We utilize that yeast platform that I talked about for immunization and single B-cell cloning as well, in addition to the synthetic libraries that I spent so much time talking about. And we also have a breadth of expertise in doing protein engineering and more specifically TCR engineering as well. And then we also have a breadth of other technologies that complement all those discovery and optimization technologies, like our heavy chain, light chain mutations for constructing bispecifics and multispecifics, as well as our CD3 antibodies for creating T-cell engagers. And then last but not least, the amazing people that I've had the pleasure of working with this last year have rich expertise in antibody engineering, protein biochemistry, computational biology, and software engineering, have really allowed us to move this field forward and have the ability to really innovate and create all of these new technologies that I talked about today. And I'd just like to close and really thank you for your time and attention today. This is a picture of us at Lake Day, which is something we do every year to really celebrate all of our amazing achievements, celebrate all the technologies that we create every year. We actually spend about 30% of our time every year really focused on technology development because we really feel that it's important for us to continually innovate within our platform. Shown here is also my email. Also feel free to come up to me after the talk if you want to talk about anything more specifically. I know it's kind of a whirlwind overview of all the things that we do at Adimab, so if you want any more information about anything specifically, please feel free to reach out. Thank you for your time and attention today.
Thanks, Aaron. Great talk. Couple of questions. First, are you using AI?
We use.
What do you think? Do you think experimental is good enough?
We use AI for predicting developability. We are trying to get into a little bit of using AI for antibody optimization. We're dabbling a little bit in de novo discovery, but we haven't really got into taking part in the competition quite yet.
I wasn't asking you about
yeah.
that. And then the second question, so you say your libraries have theoretical diversity of 10 to the 12, and you make a 10 to the 10 version of it?
So the overall the libraries themselves are 10 to the 10th, but when we synthesize them, they're 10 to the 12th. But when we put them into the yeast, they're 10 to the 10th.
But, I mean, so there is a 1% possibility that you're going to have the same antibody in two libraries, or not?
So in vitro, when they're built, they're 10 to the 12th, but when they're transformed into the yeast, they're 10 to the 10th. Yeah, so there's maybe a slight chance, but we also, at the end of the day, we also double check that we're never delivering the same sequence.
So you do the gatekeeping?
We do a gatekeeping as well, yeah.
Okay, all right, okay, thanks.
But I don't believe we've ever had a issue with delivering the same sequence.
As long as the libraries are well designed.
Yeah, we've never delivered the same sequence anyway, but I don't think we've ever had an issue with coming up with the same sequence.
Thanks. Any other questions?
Hi, Aaron. You mentioned one unique naive library per target. So if we have two targets, what's the difference between these libraries?
Can you repeat that one more time?
I see your slide mentioned per target one unique naive library.
Per target, one unique library.
That means based on target, you use different libraries?
Yeah, maybe that was a mistake. We have just general libraries that we use for all the targets, yeah.
Okay, got it. So one more question.
Sure.
Sorry.
Yeah.
You mentioned there's some affinity can reach 10 picomolar, this level. So that is based on the avidity effect or monovalent?
I believe it's monovalent, but that's most likely after doing antibody optimization. So we would probably initially do initial discovery, and then after optimization, we can definitely usually get down to that picomolar range.
Any more questions? No? Okay.
Great, thank you.
Thanks.